Thank you very much for giving me the opportunity to present a bit our work and what we are doing. And thanks a lot also for the organization team for all the support on the organization. It was really well made. I wanted to discuss a bit, bringing a bit what are the development in genomics that you can take in your everyday life and also in your research projects. Here really it's about how could we get better in genomics in neurosciences. So I'm coming from a University of Tübingen. It's in the south of Germany. I'm French, obviously, so my accent is not German at all. My institute is human genetics. It's one of the biggest human genetics in Germany, actually. We have more than 150 co-workers. We are running more than 2,000 patients discussions, and we perform roughly 7,000 analyses per year. Our focus is on rare disease, which I think in neuroscience is quite important. There is a lot of rare disease in the sector of brain disorders, but also neurology in general. We have a strong focus in oncology as well as neurosciences as a whole. Here, importantly, human genetics is really important for rare disease, as they are just so difficult to diagnose, and we discuss about that a bit afterwards. I'm working into what we call a core facility, a competence center, actually. And there, my aim is to promote development of genomic tools. We develop DNA solutions as well as RNA solutions. And in order to help the scientists to develop new methods and find the unfindable, we develop technologies such as long-read sequencing or epigenomics. I guess these are words which are very, at the moment, fashion, so you can see them a lot around it. I'm not going to discuss too much about epigenomics or single-cell sequencing, but I will introduce long-read in the talk. So why do we use genomics to diagnose disease in rare disease? Well, I mean, rare disease is really tough because many patients might have very close symptoms. However, the cause of the symptoms could be very, very different. And in the context of rare disease, with more than 5,000 rare diseases, which are at the moment annotated, is getting really more and more difficult to make sure that it's a symptom and not another. At the moment, what is very convenient is to accompany the patients through an assessment of their phenotypes, look also a tiny bit in their history, and decide whether a genetic test would help to conclude what might be the disorders. For this family, it's very difficult. Usually, a lot of these diseases are very penetrant and also very handicapping. And having a clear name on the disease as well as an idea how this disease will develop and what could help them is extremely important for the families. So usually what will happen is a geneticist will look at a case, will decide whether a genomic test will help or not. And then there are a couple of selection of tests which would be done. One of the tests which is heard a lot into genetics is this whole exome sequencing, as well as whole genome sequencing. Whole exome sequencing is basically only sequencing the part of the genome which code for proteins. This is 1 to 2% of the genomes. This is relatively cheap to do. The organization of the data is not that complicated. It's also relatively powerful, but of course, sequencing the whole genome is the most performant way of doing this. What is also important is to decide is the phenotype de novo? Is it already in the family? So in the case we have a de novo mutation, which means that the parents don't have the mutation. It's only the child who has the mutation and who has the disease. Then it's important to sequence both of the parents in order to detect the new mutation which could lead to the disease. And there, depending on if we need to do trio or not, depending on how fast we need to make the experiment, and also depending which category of disease it is and what is the affordability, one would decide between exome sequencing or genome sequencing. An important part of this workflow is the variant management. Here, the variant management is basically how to look at the data we generated in order to find what could be the cause of the disease. This is one thing which might look complicated for a non-geneticist. However, nowadays, with the software we are doing and with all the database annotation which exists, this is getting faster and simpler to do. However, here what is very important is to only call variants which are reliable, meaning it's already shown that this specific genetic mutation really leads to a disease, and also that the mutation could lead to different clinical gradients. It's very important to well data bank what are the mutation founded and what do they do to the disease. Very importantly, in genomics are the best practices. So we want to be accurate and we want to be reproducible. That's why bioinformatics pipelines, the way we are analyzing the data from genomics, are very important. And that's why also we need to have good reference standards in the clinic. And finally, something I'm going to discuss briefly is like the return of testing. It's important to diagnose disease by genomics, not only because it allows us to be more safe about the diagnostic we do and the follow-up of the patients, but also because the more genomic tests we do, the more performant we are at, not only making health records, which are helpful for the physicians, but also developing intelligent artificial intelligence, and also improving genetic counseling for this kind of disease. Here I show briefly a study we did in the context of Solvardi. I will go into that in a bit more detail, but you see on the top left that testing one single gene is like the less powerful method you could do, and testing the whole genome is the most powerful tool you could use. And when we use exome sequencing in patients with rare disease, we find roughly that 50% of the case could be solved. That could vary between 20 to 80%, depending on exactly the kind of disease. And here in our study, we had roughly 10,000 patients, 4,400 were unsolved. So what we made is, we made like the strategy one, let's try to reanalyze the data that we had with just better tools and using different tools from different universities. And out of these 4,400 which went through this pipeline, roughly 60% of them did not find a gene candidate, meaning we didn't know where to look in detail and we could not analyze further what might be the cause of the disease. In 40% of the case, we could find a candidate, meaning a gene that could explain the disease, usually 1.45 gene variant per cases, which is something realistic to look into more details. And there we basically ask specialists of the disease if they would be interested to validate the data and the variant that we found, like new variants which were never reported in the population. And out of them, roughly 30% got picked by specialists, and 120 were solved. 25 stay under validation. Some were heterogeneous variants, meaning that they could not really explain the full phenotypes, and 330 stay unsolved. So on the small diagram on the bottom, what you can see is that depending on the disease, for example, intellectual disability, they could be still well solved by this kind of pipeline. But other diseases, for example, like tumor wrist syndrome, they are still very difficult to diagnose genetically. Another strategy that we can do in order to better diagnose disease and have more cases which are solved by genetics is what we call sharing data via patient matching request. I think it's a very interesting strategy, and I think it's also for scientific work really important. In our consortium, which is called RD Connect, so it's a Rare Disease Connect. It's a portal which basically allows medical professionals to share experience and data from specific cases which could help other specialists in their field. Here, what is very handy on this portal is when a genetician finds a disease in a family, and he has a very good gene candidate, but the variant is unknown, he can ask into the network via automatic request who would have family of patients with mutation on this specific gene, which could explain the phenotypes in this specific case. On the portal, you could see that the participant ID would be the name of the clinicians. And you see that he's searching for a gene matcher for a sporadic disease at the neonatal stage for the candidate gene CHRND. So this is very important because when like cluster of geneticians find a gene which is mutated on a specific disease, they can collaborate together to also do functional testing. And the last strategy I wanted to show is, of course, one way to solve more cases is just to do more research and to also get what we call more expanded genetic events. Here I showed basically the difference between like the classical NGS, like next generation sequencing, and like the new technology of sequencing, which is called long read sequencing. The main difference between the two methods is that the sequence of DNA, which could be exactly detected by the instrument, is much longer, so a range of like 10 to 100 times longer. And this basically helps us to solve the puzzle of genetics. So using long-read sequencing, one could find new mutation which could not be found before. One thing very easy to understand is, for example, structural variation. If a gene is inverted into the genomes, it's very difficult to find the exact reads at the border of the gene, which would explain that the gene was inverted. And similarly, if a gene is moved through the genomes, it's also very difficult to find what we call breakpoint, where the genome was destroyed and where basically the gene was moved. This is much easier to do with long-read sequencing. Another example, which is very relevant for neurodegenerative disorders, but also for epilepsy, are like repeat expansion. So genomic area can have like small repeats of, for example, 3 nucleotides, 5 nucleotides, or like more complicated structures. And because these structures are typically in the range of like a couple of 100 base pairs, they cannot be detected with usual sequencing. However, with long-read sequencing, we can span the all repeats in one read and really detect in more detail in the patients if there are some repeat expansion available. I don't want to go too much in detail because I know that the audience is not like a genetic audience, but phasing is also something which is quite interesting, meaning if there is a SNP on the mother or on the father, it might be that the phenotype will be different than if the both mutations are on one allele. These are like cumulative SNPs which might play an important role into the gene function. And finally, pseudogenes can play a very important role in disease, but they are difficult to figure out. Basically, pseudogenes are genes which are different, but which have a very similar structures. And using this very short read of DNA, it's difficult to figure out if we are in a gene or in a pseudogene. However, with long-read sequencing, we have enough information to identify which exact gene are we on. So with everything said, and this is what is the focus of our labs. The focus of our labs is to resolve complex rearrangement into genomes. Our strategy is summarized into the paper I put it there, and the idea is very easy. The idea is that genomes is a lot of information, and putting all the genomic information together to find modification of the structure of the genome is almost impossible on the normal computer clusters. However, with smart design and with good algorithms, this is still possible to do it in high throughput for a large amount of patients. The strategy we use is we use NGS, short-read sequencing, where basically instead of having a genomic region of interest with a read 1 and a read 2 pointing toward each other, with like an insert of 300 to 600 base pair, there is some anomaly in this read. For example, instead of pointing one to the other, they are like inverted, which seems to mean that there is an inversion in the genome happening. Or what we call a translocation. A specific read is on a chromosome, and another read is on another chromosome. That means that 2 chromosomes got reassembled. Or a deletion between 2 specific reads. There is a much longer genomic region than anticipated. So when this is detected on short-read sequencing, we can do then long-read sequencing and try to validate the events. This could be done looking at this kind of issue. But that could be also done looking at how we discover specific genomic region. And with our algorithms, the idea is to find the exact breakpoint where is the genome broken. And these algorithms work very well. So here in this example, we use a cell line with a lot of genomic events, so more than 800 of them. And what we could detect in this cell line is that using our pipelines, this kind of calculation could be done in a couple of hours, and all the single event could be validated using different technologies. So these methods are very powerful. They run very well on limited hardware. They can be run with very large cohort of patients, like several thousands of patients in a reasonable amount of time. And they are also very robust. Now going to the RNA topic. The problem of this structural variant is we don't always know what they do in the genome. One could see that the genome is broken, but if that would lead to a phenotype or not is another problem. So typically what we propose at the moment in our institutes is patients who come for consulting, genomic consulting, will give blood for DNA isolation and for RNA isolation. And this is for us a very powerful strategy because we can get from one visit DNA and RNA, and we developed method for storage of DNA and RNA in order to process the sample stepwise. Importantly, what we learned from research is this kind of tests, they need to be done with a very specific quality and with a very specific reproducibility, and this is very difficult to reach without lab automation. Therefore, at the moment, what we do is we automatize RNA isolation on a platform, and we automatize the generation of a library for sequencing on a second platform. And that allows us to perform a couple of hundred of samples within a few days with very low variability independent of the user, as well as a relatively simple protocol with reduced cost. One of the first methods that we established in our pipeline was the validation that variants of coding region. And this really matters because not all the mutations are all annotated. That means that, for example, if we take a blood sample with 25,000 genes which are expressed, we can commonly detect 15,000 of them, so almost like more than half of them. And if we find, for example, a variant which is not known in the database, but which have a function which is common to another mutation, for example, a mutation on a critical domain, on a regulatory domains, and so on, we could test at the RNA level that it's not loss of expression, that it's really a change of the RNA and of the protein. So this power a lot our analysis, especially in rare disease where a lot of variants are still not annotated and where it's difficult to make a diagnostic. Something else that we do that I think is also relatively easy to understand is we also test with the RNA if the splicing of the RNA is correct. In a normal case, for example, we would have 3 exomes. There is an exome gray, purple, and gray, and the splicing can be with or without these specific exomes. However, what could happen is due to mutation in, for example, deep intronic regions, it might be that the splicing does not happen anymore. So a new exon is spliced in, an intron is retained, or an alternative splice site would happen. And again, these mutations are usually not annotated in database. And if we find them in whole genome sequencing, we can look directly in the RNA sequencing to see whether this mutation that we found they might play an important role into this protein or not. So that basically allows us to speed up the genomic testing by building hypotheses and testing immediately with RNA-seq whether these hypotheses are correct or not. And for us, what is really important is we manage to integrate this analysis into a tool which allows us to test is there on a gene specific deletion insertion which could break the splicing, and on top a filter which allows us to detect automatically whether splicing was indeed changed into this specific gene. So that simplifies a lot the work of the analyst. Something I want you to discuss very briefly is issue of gene expression. The problem of gene expression with RNA-seq is that the gene expression is not stable in individuals through the time. That means that it's very difficult to generate a database of blood where we can identify very easily what we call outlier, meaning genes which are supposed to be expressed, which are not expressed, and vice versa, which are not supposed to be expressed but are not expressed. And there are many algorithms which are available in order to do that, but here what is critical is the metadata of the patient. So here a warning, I think at the moment gene expression analysis of blood in large cohorts which are not aimed to be compared is still technically challenging. And the final application I wanted to bring also because as I mentioned, we do quite a bit of oncology. RNA-seq allow to also detect fusion genes. That means that if there is a large deletion in the genes, and we expect that, for example, 2 genes will be fused into one specific messenger RNA. RNA sequencing can help us to figure out whether this specific gene was fused with this one. And this also saves a lot of time when we do diagnostic to directly validate that this fusion gene exists or not. A very short example of a study that we made is we found in a family of patients a stop loss variant. That means that the stop codon of a specific gene was lost. And we wanted to detect at the RNA level whether there is a non-mediated decay, which is reported in this case to leads to a phenotypes, or whether the stop loss is indeed leading to an expanded protein, which leads to a disease. As the mutation was not known, it was not clear for us if a mediated decay will occur or not. So we took fibroblasts from these patients, not blood in this case, because blood was not expressing CLDN11. And by sequencing the RNA of an individual versus a control, we detected the mutation by the arrow here. And basically, we could validate that the protein is expanded, and leads to the specific phenotypes. So my take-home message is if you have course of patients where you would like to make genomic tests, collect blood for DNA, but also collect blood for RNA. This will save time into your study, especially because you can store this tube for long term. You don't have to analyze them immediately. And that will really boost up your yield of diagnostics. One application which is easy is aberrant splicing. This is something which could be done relatively easily, and the loss of heterozygosity is also something which is relatively easy to test. However, the analyses which are based on gene dysregulation, they require a very large data background. They also require a lot of metadata of the patient. Would like to acknowledge the team of Professor Rees in the institute where I'm working, as well as all the scientists and technicians who are involved in these projects. And thank you for the opportunity to presenting our research.